So yes, today we will talk about a couple of sources from the CGW knowledge base, practice guidelines, which we've also referred to sometimes as treatment guidelines, and clinical trials, how you can view the content and use it in your reports in CGW. So here's our agenda for the day, for this 1 hour session. I'd like to give a brief overview of the CGW collaborative knowledge base to put these sources that we'll discuss in more detail today into some context, and review some content that was covered by Will at our last webinar in October, I think, regarding automated variant classification, to make sure we have proper context for the rest of our discussion today. Then I'll talk about how our annotation works of these specific sources of practice guidelines and clinical trials, so that you understand where we get this information and how we handle it and how it winds up getting into the knowledge base. Then we'll look at a few options of how our current clients are using using these sources to populate their reports, how they're displaying this information. And then finally, we'll do a bit of a live demo in the variant details page of CGW to look at how to search for trials and guidelines and find the most appropriate content to include in your reports. Do please interrupt me with questions, or I suppose type them in, and Sarah can interrupt me if you have something you wish to ask. I'll be pausing in a few spots if I remember to allow you to allow ourselves to answer a few questions regarding background info before we dive into the more detail. Okay, so we'll start with a bit of background information. So many of you will be familiar with the CGW collaborative knowledge base and the sources that we annotate and pull into the knowledge base. The breadth of the knowledge base is represented by our variant details page, which we'll look at later, and with which many of you are familiar. We have sort of a broad range of sources, both publicly available and some that we curate to some extent and some that we curate heavily that are proprietary sources, as well as user-curated content. That's the shared interpretations that users of CGW write and save to the knowledge base. These span a range of clinical evidences as shown in this slide, where at the bottom we have public sources and in silico evidence. This includes genome builds, annotation releases for genes, transcripts, proteins within those genome builds, pathways, population frequency databases, and computational predictions. All the way up to high levels of clinical evidence. Above this dashed line are the sources that have the most clinical evidence that really support definitive classifications that can be automated in CGW. And so those include, at the top, the shared interpretations that users write and save to the knowledge base, the practice guidelines and FDA drug labels, which we're sort of combining into the category of practice guidelines for our discussion today, as well as clinical evidence from sources like ClinVar, and aggregated patient data from past patients' cases analyzed in CGW. Trials then falls beneath this line. We don't use those trial information that is ongoing clinical trials that are recruiting in order to classify variants, for instance, but they're a source of information that your oncologist customers may be interested in seeing on the reports you return to them. All right, so that's a bit of a background on the knowledge base and where those sources fall that we'll be focusing on today. The classification scheme for somatic cancer assays, any of you already in production will be very well familiar with this. It's a 5-level classification scheme where levels 1 and 2 are actionable categories where there's some predictive, prognostic, or perhaps diagnostic information, either in the malignancy of the patient exhibits or another malignancy. That'd be levels 1 and 2 respectively. Level 3 is a variant that may not have that level of supporting evidence for action, but has been described previously in cancer, for instance, in cosmic, or in ClinVar, or in some other disease. Then I'll skip to level 5, where there's evidence that the variant is found commonly in the population and is not likely to be a disease-causing variant or pathogenic. And then level 4 is the catch-all for VUSs, where there's no other information in any source in the knowledge base. And so, as Will would have described to you in the webinar he gave, many of the sources in the knowledge base can automatically classify variants into these categories based on where they are found in databases and our own curated sources. And so just to review those sources of sort of basic classification rules that most of our somatic panels use. First is interpretations that were written either by your institution or other institutions and that have been published to the knowledge base. Those take priority and precedence over other sources of classifications. Second are practice guidelines / drug labels. These are the sources we'll be talking about today from NCCN, ASCO, and FDA. These can classify variants into levels 1 and 2, whereas interpretations can actually classify variants into any level depending on how they are written and what level of classification is associated with the interpretation when it's saved. And then third, as we described on the last slide, are other databases, population frequency databases that could drive variants into level 5, or ClinVar Cosmic, which in some classification rule schemes could cause variants to be populated into level 3. And then we can write other custom rules per your specifications to put certain variants that you feel should be level 5 into that category, etc. So before I go further, are there any questions about that background information that anyone's got? This should be very familiar to those of you who are already using CGW on a daily or weekly basis. Okay. So let's look, before I get into too much detail, at some basics for practice guidelines. What are we talking about here when we talk about practice guidelines? So these are both, the way we group them are both practice recommendations from the National Comprehensive Cancer Network, or NCCN, and from the American Society of Clinical Oncology, or ASCO, as well as regulatory approvals, that is FDA drug labels. And so this is for the use or avoidance of use of a drug, so a known responsiveness or non-responsiveness. Could also be for prognostic information, less so from FDA, but more from ASCO and NCCN, or even diagnostic information. So all of those management impacts, if you will, are present in our knowledge base. The impact of these practice guidelines to your reports in CGW for your patient cases are, first, that these rules, guideline rules, that is logic written in CGW, as we described, can cause those variants to be automatically classified as level 1 or level 2. And whether they're 1 or 2 is dependent upon whether there is a disease match between the patient's disease and the disease with which the annotation was written, the disease level at which that rule or logic was associated. Also, depending on your desires for how you want your reports to appear, you have the option of having drug information or prognostic information automatically populate into your report at report generation, which it can then be edited and changed as you wish on a case-by-case basis. We'll see some examples of how this is done right now in CGW a little bit later. we'll get to this in the live demo, but there are some useful parameters on which you can search or filter within the variant details page in CGW to uncover the most useful or interesting treatment or practice guidelines. Those include the source, that is FDA, NCCN, or ASCO, the disease with which the annotation is saved, and the annotation level, which I'll describe in a little bit. That's this annotation specificity level that we record as well as we do annotations. And as an overview for trials, our current sole source for clinical trials right now is clinicaltrials.gov. We annotate oncology trials from that source. As I described, trials do not impact variant classification. However, they can populate the report automatically if you've chosen to have that in your reports. And so again, we'll see some examples of how that can work. Again, as you are reviewing this content in our knowledge base, it's useful to filter clinical trials on disease, on phase, whether it's a phase 1, 2, 3, or 4 trial, or something in between those, annotation level, again, and then geographic location. Okay. So now, how do we take these sources and annotate them to be discretely represented in our knowledge base? So here's some basics that apply both to guidelines and to trials. These annotations are performed based on searches of this content for a set of genes and a set of drug names. So we annotate now something on the order of 500 plus genes in oncology trials and in treatment guidelines, or rather practice guidelines, based on the genes that CGW users are reporting on their reports. And so this happens sort of in two, you can think of it in two different approaches. One is the initial annotation when we add, for instance, a new gene to the list of genes that we annotate. We do a search across that source, identify all mentions of that gene or drug, if it's a drug that we've added to our drug search list, and then perform annotation from there. In addition, we are performing weekly annotation, or rather weekly searches of changes to these sources. So NCCN puts out flash updates whenever any of their documents are updated or new documents are released. Likewise, we do searches of ASCO and FDA and clinical trials to find what has changed since the previous week. All right, so we're keeping up with these on a weekly basis, and then moving those changes, additions, removals, like trials that are no longer enrolling, to or from our knowledge base. Okay, so one concept that we've introduced or expanded, I guess, in the past year or so is the level at which we annotate findings or claims or trials in these sources. So these are represented in the annotation level column in the variant details page, which we'll see. The first two levels are variant level and gene level. All right, and you'll see I've got my spectrum here, specificity, and this gets to that specificity of annotation that I referred to a while back. So the most specific annotation approach is at the variant level, where a specific variant syntax is referred to in a source. So, KRAS G12D is explicitly mentioned in the trial or in the treatment guideline or FDA label. We'll give some other examples here as we go as well. One step down from that is the gene level specificity. And that is where there's no specific variant syntax mentioned, but the gene name is given, so there's no ambiguity there. And in some cases, there will also be some more general information about the type of mutation. So for instance, a FLT3 internal tandem duplication, which can be a lot of different size insertions. So it doesn't have a specific syntax, but it is relatively specific in terms of how to annotate. KIT mutations in exons 9, 11, 13, whatever those are, or a sensitizing mutation in EGFR. We can figure out from that, in fact, based on NCCN guidelines, if it's a trial that we're annotating, what they really mean by that. And so we can do some more specific annotation there so that we're not throwing a net over the whole gene or any variant in the gene. And so that we would label as a gene level specificity annotation. There will be times when a trial, for instance, is so non-specific as to say, any RAS or BRAF mutation. And all we have to go on there is, assuming that it's a non-synonymous change in those genes. So that's maybe the broadest, least specific annotation that we store or annotate with the gene level annotation. Importantly, treatment guidelines loaded on production today are all at these two levels, variant or gene. Okay? Now we do have less specific annotation levels. We recognized some time ago that when we searched simply for gene names, for instance, across clinicaltrials.gov, we missed a lot of trials that had useful information that we could infer would be possible trials to consider in the context of certain mutations. And so we began doing searches based on drug names that we can associate, that are known to target specific mutations or specific genes. And so there are two additional, less specific annotation levels that we annotate, and these are currently on production for clinical trials, unlike guidelines which are limited to variant and gene level. All right, and so the drug level annotation means that the gene is not referenced by name, but there is a drug in that trial that is offered, and we know, based on NCCN or other sources, that this drug targets a given gene. And so we can then make a claim about making association between a mutation in that gene with this trial. And then it would show up as something for consideration to put on the report in the variant details page under the clinical trials tab. Now, to further extend that, the pathway level of annotation is selected when a gene is not referenced by name, again, but there is a drug that is referenced, and that drug targets a pathway of which a particular gene is a part. So a good example here is an inhibitor of mTOR could be annotated for mutations in PIK3CA because PIK3CA is in that same pathway, and an activating mutation in that gene might be a tumor with that mutation might be responsive to an inhibitor of mTOR, even though that drug does not act directly on the gene that is mutated. So the purpose here, again, is to be able to increase the amount of content that we can display that might be of interest, but it's not as specific. By definition, it may need some additional review rather than automatic population integer report, for instance. Okay, so let's look at some specific examples here for treatment guidelines. First, let me just give you a brief look at the sources for treatment guidelines, specifically kind of what the documents are that we start with to do our annotation. Here's a snapshot here of the variant details page where this is for an NRAS Q61K variant, where you have this is just page one or part of page one of the returned results, where you can see multiple sources here are returned. And again, you're familiar that you can customize which columns are shown here, but you can see the annotation level is shown and the impact of the management impact or drug impact. This is from our test server, by the way, so that's why you have a drug level annotation that shows up. That's not loaded on production today. The FDA drug label is this PDF document that we can search for and download. This is just a snippet of what's shown. You can see this is regorafenib, which is demonstrated to be a drug that can be useful for tumors with RAS mutation, activating mutation. So we search through this document. It's returned to us via an automated search, but then our annotators do manual review of these in order to essentially turn the PDF document text into something, a discrete rule in our system that can, some logic that can fire. Likewise, the NCCN treatment guidelines or practice guidelines documents look like this. They're a big long PDF with a section at the top with a specific algorithm that defines some useful information, and then a long discussion section looks like this, which from which we then can look into specific cited references and then draw inferences and make claims about responsiveness, non-responsiveness to specific drugs or prognostic information. ASCO treatment guidelines or practice guidelines come from basically from articles written in JCO. These are clinical opinions or provisional clinical opinions. So we are, again, searching these documents as they come out in order to pull out the useful information and create additional discrete logical rules that can fire and infer content in your reports or within the variant details page, at least for your review. So for these practice guidelines, we have our list of genes, we have a list of drugs that are useful for annotation, and we perform an automated query of these sources in order to get hits returned that can be reviewed by our annotators. Again, this could be maybe a brand new gene we've added where we're looking across all the documents to find any mention of that gene. It could be a weekly update or any updates that we are notified of from NCCN. But now we're going to see what changes have been made to these documents, and within those changes, are any of these drugs or genes mentioned? So the context of that PDF document is read by our annotators, and they also look at reference publications and read those, and then they create specific annotations based on those publications or that practice guideline document or drug label. All right, so here's an example of, this is a really oversimplified examples I'll show of how these rules are written. There's basically a condition. So this includes, among other things, the disease, the gene, the variant. It can also include the consequence, coordinates, genomic coordinates, type of variant, if it's a copy number change or a single nucleotide change. These are all possible conditions that can be annotated. And then the annotation content itself, again, this is very oversimplified, but you have a source, a drug, an impact, and additional information, text or reference claims, all sorts of things. So here's an example, simple example of a BRAF V600E mutation in malignant melanoma, where FDA, as well as ASCO and NCCN, surely recommend or have shown approval for using vemurafenib, that mutations, rather tumors with these mutations, are responsive to that drug. So this is the same example we just saw. This is an example of a variant level annotation, right? This is a very specific syntax. The document says that it's, this particular amino acid change is what confers responsiveness. And again, there could be others. There could be other BRAF mutations that are likewise responsive, and those would be annotated as separate rows, separate rules, but could fire very similarly. As an example of a gene level annotation from a practice guideline, here we have an ASXL1 annotation in acute myeloid leukemia, and here we have no specific variant syntax. We have the consequence of non-synonymous. So any non-synonymous change in ASXL1 could fire this rule where it's shown that NCCN doesn't have any drug information, but this is associated with unfavorable prognosis in the context of that disease. So again, that is less specific in the sense that there's no particular mutation that is noted, so we have made an inference, a logical one, that non-synonymous changes in that gene are those that's the context in which we want this content to infer and this to appear in the report by default if you've configured your report that way. And then just to extend this a bit, here's an example of a variant level annotation that has two sets of conditions. So we write a number of logical rules that operate in this fashion where more than one variant must be present or more than one condition must be met for that annotation to appear. In this case, we have, in the context of CML, chronic myelogenous leukemia, two ABL1 mutations. One is a rearrangement with BCR, a fusion, and then another is the T315I variant. And so in combination, NCCN says that such tumors are, or such leukemias are responsive to ponatinib. All right, those are just some examples, again, at the variant and gene levels. I'll show you some drug and pathway examples in the trials annotation. So, do we have any questions we want to pause for, Sarah, that you see, or shall we plow ahead?
No, I think we're doing well.
Okay. For trials, again, we are searching oncology trials on clinicaltrials.gov. And so here's the trials tab for this same variant details page. And I'm showing here a set of columns, including our annotation level, some information about drugs, inclusion criteria, etc. Again, these are all configurable as you're aware. So for trial annotations, we query again by our gene list or drug list. Trials that come back as hits here are screened by annotators, and then valid trials are annotated. Trials that are not enrolling currently are annotated but sort of kept out separately and only added to the load in production when they are in fact enrolling. Enrollment begins, and it becomes an option for an oncologist potentially to put their patient on that trial. So here's an example of a variant level annotation for a trial. We have glioblastoma, IDH1 mutation, R132H, and here's a trial I found. It's a phase 2/3 trial in Phoenix of a particular agent. I've obviously greatly shortened the trial name, but this is a very specific syntax annotated trial. At the gene level, we have an AML trial that says that basically, I think it's PDGFR-A and might also have been KIT mutations. It wasn't more specific than that in terms of any particular mutations in those genes. There's a phase 2 trial for Philadelphia chromosome negative AMLs with this particular agent that I will not attempt to pronounce. And so this is an example of a gene level annotation. And I've just shifted those up to the top to make some room for ourselves. A drug level annotation here from a trial I found for NRAS. This is for colorectal cancer, a regorafenib trial in phase 2. And so this was to, I think, identify a biomarker that predict responsiveness to regorafenib. And so this didn't actually talk about NRAS in the trial itself. It talked about the drug, but we know from our knowledge in the NCCN guidelines that NRAS mutations are responsive, can be responsive to regorafenib. So this was added as a trial that could show up if an NRAS variant is identified in a patient. And then the final extension there is the pathway level annotation where, and this is really the same example that I gave. This is a trial for breast cancer where they're adding everolimus to hormone therapy, and this is a drug that targets as an mTOR inhibitor, and so this could be annotated potentially for mTOR as a drug level annotation, but for PIK3CA, which is in the same pathway, but is not itself targeted by this drug, we've annotated this as a pathway level annotation. So this is all there, again, for you to be able to have more content at your disposal and then be able to tell at what specificity level was this content annotated, so that you can, apply more scrutiny to those that are on a drug or pathway level, because we've had to make some inferences about what the trial really intends to target, because they were not very clear about specific mutations or genes that they were looking for mutations in for enrollment. And perhaps they are not, but it is something that is without this approach, we may be missing some content that is useful for reporting. So questions now about the annotation approach, anything about annotation levels that are unclear. You may recall that we sent an email maybe a couple of months ago indicating that we had rolled out the drug and pathway level annotations for trials, and so those are now available in production. We have not done so for treatment guidelines or practice guidelines to date. We need to develop a way in which we can keep the drug and pathway level annotations, the responsiveness statements, et cetera, off of the reports by default. We want to keep the default populating content to the most specifically annotated, the most secure content for variant and gene level, and so that's where we have that for today. We do intend then in the future for drug and pathway level annotations for treatment guidelines or practice guidelines to not impact classification, not show up in the report by default, but be in the variant details page for your review and for you to be able to promote to the report should you choose to, or to write about in your interpretation. Okay, so let's move on then to looking at some examples. I've taken some screenshots from reports that give you an idea of, if you're not familiar already with what our options are, or possible options are, for showing some of this content on your reports. So the result summary section of the report is intended to give a high-level snapshot of the key findings in the patient's tumor, and this is one of our formats for display of this content, where one can show essentially corresponds to this column level 1 variants, level 2 variants, or level 2 related findings. Here, showing responsiveness or non-responsiveness to particular agents. In this case, in another malignancy other than the one in the lung adenocarcinoma that was accessioned here for this patient. And then additionally, prognostic practice guidelines can be added to the result summary section. Clinical trial opportunities can be indicated as well, so you could choose to include variants that may not be actionable per se in this section. And then for those of you who have used a report template that has this content on it, these drug names or the disease name for responsive, or rather prognostic or diagnostic statements, can be edited in line here manually, so you can make some changes there. Likewise, the statement about presence of a clinical trial opportunity. And then in a newer version of this section of the report, this result summary section that maybe a couple of you are already familiar with, we've added the ability by clicking on this plus icon in a given cell in the table to manually add a statement about responsiveness, prognosis, diagnosis, or even risk. And so should you find that you don't like where something is automatically populating in terms of which column, or you want to add another statement that from some other source you found, or that you weren't able to promote directly from the variant details page for the given variant, you can add that manually. So this adds some flexibility. If you're interested in being able to do this and you don't have that capability now, then do talk to us. Now, then moving down to what we've traditionally called our clinically relevant results section, right, where in addition to a high-level summary, we can add content like interpretations and more prose that you might write to describe the meaning of this variant to this patient's disease. We have developed some tabular displays where first for this treatment summary table, as we've called it, we can show a list of drug names to which this tumor may be responsive or non-responsive, or in these cases of these two rows here, to which there may be a drug option that's available through a trial. We haven't made a claim about responsiveness or non-responsiveness, so you can actually toggle that icon to something else. But this can serve as a high-level summary to the oncologist of the the available drug options that they may want to consider. Again, this is optional content that you can include in your report template if your customers, your oncologists that you serve, want you to provide this information on the reports. Again, some do not, and so we have our users really run the gamut there of leaving out all drug information and trial information all the way to really wanting to maximize the content. And so this one was developed to show us maximize the treatment, the drug and trial content. And then down here, this table again is on a variant-specific basis. So we've kind of created this set of tables for each variant that was deemed worth talking about or actionable in some way. And one can add and remove trials. One can have certain rules to have trials populated only within their state, only within certain phases, only for certain diseases, etc. And then, of course, manually one can change what shows here on a case-by-case basis. So these are some of our newer options for how to display this content in a report. Now, some of you will be familiar with this simpler display of clinically relevant results, where all variant content, be it interpretations or trials, appears in a single table. One can simply write in information about an NCCN guideline that is relevant to this variant and make that part of their interpretation. It's another means by which you can incorporate this content, albeit less automatically, unless this is an interpretation that you've written previously and saved, and you want to automatically populate in the future for a particular disease or malignancy. There's another approach. And likewise, trials can be included in this sort of clinically relevant results summary table. Okay, so that is the extent of the slides I have for today. Since we have some time, and do we have questions that we should cover before I move on? All I intend to do beyond this, other than fielding questions, is to spend a little bit of time in the variant details page reviewing some strategies for when you have 80 trials that show up in the variant details page, how to whittle those down into those that might be the most appropriate for including in your reports.
Yeah, let's go through the workflow in CGW and then we can come back to questions.
Okay, that sounds good. All right. All right, is this showing up okay?
Yes.
Great. All right. Okay, so I'm on the guidelines page, practice guidelines for a BRAF V600E. So if we take off all of our filters here, we have NCCN, FDA, we have 64 results here for treatment guidelines. And again, because we're on the test environment, I do have a couple of examples of drug level annotations, but these are not available on production at the moment in order to avoid any non-specific content appearing into reports by default. So an approach here for looking through guidelines and deciding what you want to include, again, depending on your report, some of this content may already have auto-populated, but you may have removed some of it and want to start fresh, or you may have a report template that doesn't necessarily have this content by default, and you want to review it yourself and put it into your interpretations. You can filter on source. Here we don't have ASCO in this example, but we have FDA. Annotations, that gets us down to 12. And then the annotation level is another useful filter. So I would recommend starting at the variant level and finding the most specific practice guidelines that have been annotated and working with those. You see that about half of the FDA annotations are at the variant level, where there's specific responsiveness statements for these various agents. And you can always then click on any row of any of these tables or most of these tables in the variant details page for most of the tabs. And rather than having to add 100 columns to see all the content, you can simply scroll through here and see what's been annotated. Not all the fields will be populated because they're not all necessarily relevant for the particular annotation. We have pulled several sentences from, this is either from the actual NCCN practice guideline PDF document or from a paper that was cited in that document where there's particular information about what a given study showed and the supporting information here as to why this annotation has been made, or why this claim has been made about responsiveness. Then there's often going to be there'll be a link to the document itself from NCCN, which is behind, sorry, this is FDA, pardon me. The NCCN documents themselves are, I believe, behind a paywall. So unless, no, that's not correct?
No, you can create a login on your own and then...
Okay, great. Great. So you can access that directly if you want to read more. Likewise with ASCO and FDA, all of them have link outs. I'm not showing that column right now to save on space, but that is something you can do to dig in further. If we look at the, I think it's the drug level, we have a small number of annotations. Let's see, do we have anything at the variant level? Or rather, gene level? We do. So a couple of gene level annotations, less specific, may not cite a specific mutation, but something about BRAF mutations or activating mutations, and then likewise down the line to drug and pathway. Now for NCCN, you might want to go to the variant level. What do we have? 50 results still, so you might want to look at, perhaps you want to add drug impact and look specifically at non-responsive statements, drug impact statements. That's 19 of the 50 results. So there's additional levels of filtering you can do to whittle down to those most useful treatment guidelines. And there is some level of duplication here where there may be multiple significant patient outcomes. There may be multiple rows shown. So that's something to be aware of. You can get rid of that to some extent by doing some additional filtering. In clinical trials, I find it useful to filter by phase, as I showed on the slide previously. So, ideally, you can identify a higher phase trial that's already been through some vetting in human subjects, and identify phase 3 or phase 2 trials. Also, the annotation level will come into play here. So those at the variant level are perhaps the most, certainly the most specific and perhaps the most relevant. And then, you may really want to go to the level of, is this a trial in France? Maybe I'm not interested. And this is a free text search for the moment, so maybe I want to see if there's anything here in the St. Louis area. It looks like there's some trial or other in Missouri where there's a site in Missouri. So that's one way to get down to a trial that might be more accessible to your oncologist patient if you can pin down that most of the patients are likely to be from your area. I should note that this geographic restriction can be applied to a report template so that automatically you only get trials that are within a certain region, within certain states, etc. So that's an option. So from here, I might stick with the higher phase and move to gene level and see if I get any more hits that are within my region. I've only got one trial there, and so on. You might also want to limit it to drug, or rather by disease. So this might be a malignant melanoma. By default, the inferred practice guidelines don't limit by disease. So you get any practice guideline that is relevant to that mutation, and then it'll be level 1 or 2 depending on whether there's a disease match. But if you want to whittle down just to those, sorry. That are relevant to malignant melanoma, then you can do so, and it might require, removing certain filters to find examples. Here we go. Phase 1-2 trials that are at the drug level. There's a couple of those, or certainly if I open this up now, I should get quite a number. Yeah, looks like I do. 67. Okay, so those are a couple of strategies that I think our variant analyst team. We have a team of folks who are also part of our annotation team who can do review of reports and preparation of interpretations and reclassification of variants. That's a service we offer. I asked them for some input on how they go about identifying trials and guidelines for consideration, and these are some strategies they use in order to find the best content. And so your mileage may vary. You may have your own approaches, and certainly interested in hearing what you found useful and solid approaches to identifying the best content. And over time, we can add more automatic logic that can use those strategies to really pull out the best stuff and put it onto report templates automatically. So we have only 8 or so minutes left in the hour. I think I'll stop here and open it up for questions. I guess I do want to make sure to mention the next webinar. Shall I do that now?
Yes.
All right. Let's see if I can get back to it. All right. So our next release of the application, CGW 6.0, is due out on test later this month and then production in March. So in February, we'll be doing a webinar to demo the new features of CGW 6.0. Those include support for added support for constitutional exome, including automated, at least partially automated ACMG classifications. Oh, well, I put the wrong date on there. It's 2017. It is not in the past. So this is coming up here in about 4 weeks. The ACMG classifications is a big feature of 6.0. We also are rolling out clinical data aggregation, where we are showing information, in a de-identified fashion across all past cases run in CGW to give you some indication of the frequency with which variants that variant appears in certain tumor types, and then finally some additional features that we have incorporated after our merger with Toot Genomics that happened a few months ago, where we're now incorporating some of their features, including their phenolizer service, which is great for identifying likely pathogenic variants based on a phenotype-based workflow for constitutional exome, as well as being able to filter based on inheritance models. So those of you who are interested in constitutional will be happy to hear of these new features. All right, with that, let's leave the last few minutes here for questions. Thanks very much.
So the first question we have, Andy, is whether it is possible to see the level of annotation, whether it's variant, gene, drug, or pathway in CGW.
Mm-hmm. Sure. So at the moment, the place you see that is in the variant details page. Okay? So there's an annotation level column. Right now in production, that column is not a default column, and we're going to make that change so it appears. It's showing here, but I've added it before our webinar.
Okay.
So
maybe
yes.
we can demonstrate how columns are added.
Oh yeah, absolutely. Sure. So here, under this customized columns, pull down, drop down menu. It will show you, as if you have no text in the search bar, all the checked boxes are those columns that are already showing. And then from there, one can do a search and add additional columns. One can also simply browse through, if one wishes, the available columns and check off any of interest. And likewise, any of these columns that you can add here can also be filtered on. Okay, so I think we've gotten rid of our phase filter here, but you can add phase, you can add any of these. If you have a trial ID you want to go look for, you can do that too. So anyhow, the annotation is here. You can both filter and sort on it. Sorting will be alphabetical. Filtering will be more specific about, you can check off the specific permissible value, that is one of the 4 terms that are allowed in that field. And that's available now for clinical trials and for guidelines. The other sources, the annotation level doesn't really apply. Other questions here?
Yes, there's one more question. It says, if practice guidelines are not restricted to a disease, then how does the system decide to assign a variant a classification level of 1 or 2?
Mm-hmm. Sure. So the practice guideline annotation does have an associated disease, right? In this case, we're in trials, but we're looking at most of these for BRAF, not surprisingly, are written on. Well, hang on, these are not, in fact, malignant melanoma, but there are plenty for colorectal cancers you see here. And so, essentially, what CGW will do is try to match the disease of the patient to the disease of the annotations. And so, if this is a patient with malignant melanoma, and the only annotations here are for colorectal cancer, then those guideline annotations will drive the variant into level 2 because there's not a disease match there. Of course, here for BRAF, we've got plenty, certainly, that are written for malignant melanoma, and so CW will look at all the evidence and pick the most severe classification. It will match the malignant melanoma disease from the patient to the annotations for the matched disease and call it level 1. So I should be clear that all of the FDA, ASCO, and NCCN guidelines are annotated at level 1. That their default is level 1 if there's a disease match, and then if not, it becomes level 2. Just like the other rule disease matching, for instance, interpretations, where a level 1 interpretation for lung cancer can show up in your colorectal cancer in the report template, but it will, because it's not a disease match, it will infer as a level 2 variant. Same story with your treatment guidelines. If there's a match, it'll be level 1 or 2 depending on the disease match.
Okay, I think we're almost out of time here, Andy. Thank you. Thank you, everyone, for attending. Please stay tuned. You'll be receiving an email soon about the next webinar that we're having in about a month's time on February 9th. And please help provide feedback on these webinars if they're useful to you, and if there are other things that you're interested in hearing about from us. Let us know, and we can incorporate some of those suggestions into our webinar content for future months. Thank you all, and have a good rest of the week.